So first I just want to say thank you to Mike for the really kind and generous introduction, and also a really big thank you to the ASBMB Lipid Research Division and lipid community for their support and for selecting me for this award. This is truly a huge honor to receive this award. So my lab is interested in the cell biology of lipid metabolism. We're interested in ER-derived neutral-lipid storage organelles called lipid droplets, how they form under different metabolic conditions, how proteins localize to these organelles, how the proteome is remodeled by systems like the ubiquitin proteasome system. We're also interested in how lipid droplets make contact with other organelles and communicate lipids in order to maintain membrane homeostasis. For the sake of time, though, I want to focus on a different area of the lab, which has been developing, which is around lipotoxicity, how our cells maintain lipid quality control, and are the defenders against a form of cell death called ferroptosis. So really, the major question I'd like to think about today is how is lipid quality maintained? And our cells really sit at a precipice. At any given time, they have to combat the potential accumulation of damaged biomolecules that can be cytotoxic. And fortunately, we've developed highly sophisticated quality control systems to do this. We have preemptive quality control systems that suppress this damage. We have repair quality control systems that repair this damage. And we can also simply degrade or metabolize that damaged molecule into a non-toxic molecule. I think this is really well exemplified in the form of protein quality control, where we can have misfolded proteins, damaged proteins that are prone to aggregation and cytotoxicity, but we have chaperones. We have the ubiquitin proteasome system, we have autophagy lysosome system, and we have homeostatic stress responses like the unfolded protein response, or UPR. But what about lipids? How are their quality control maintained? And I'd like to propose that similar to proteins, we also have to have quality control systems. And indeed, we know that lipids can be prone to damage. For example, polyunsaturated fatty acids, or PUFAs, can be oxidized, and this can lead to defects in viability and cytotoxicity. So what are these quality control processes? So this question led us to become interested in this form of cell death that you may have heard of, ferroptosis. This is distinct from other types of cell death like apoptosis, but for us, this was really an exciting area because it's a form of cell death that's a regulated type of cell death due to failure in lipid quality control. So this directly connects lipid quality control with cell viability. So in this form of cell death, we have the incorporation of PUFAs into membrane phospholipids, and then in an iron-dependent manner, the generation of lipid peroxides. If these accumulate to high enough amounts, this can result in membrane rupture. Now, there's a well-known quality control system that defends against this. This is mediated by GPX4 and has been characterized by Brent Stockwell and others. And this system utilizes the glutathione, an abundant antioxidant, as a cofactor to mediate the conversion of lipid peroxides into non-toxic lipid alcohols. And there are some useful small molecules, these developed by Brent Stockwell's group, for studying ferroptosis, such as RSL3, an inhibitor of GPX4, and erastin, which results in a defect in the synthesis of glutathione. Now, this form of cell death has really exploded in recent years due to the potential therapeutic importance, with the idea that perhaps we could trigger ferroptosis as a way to kill therapy-resistant cancer cells, or that we could inhibit ferroptosis as a way to ameliorate degeneration associated with a wide number of diseases, including neurodegeneration and ischemic tissue damage. Now, remember when we got interested in this, we certainly could see that some cancer cells were very sensitive to GPX4 inhibition, but others were very resistant. And in fact, in some, we could delete GPX4, and these cells would continue to grow and thrive in vitro. And this really suggested to us that there must be additional protective mechanisms that compensate for the loss of GPX4. And in work from postdoc Carol Brazuker and Zhirpong Li, we took a genetic approach to identify these compensating mechanisms. So we performed synthetic lethal CRISPR screens, in this case, introducing a library from our colleague Mike Bassick down at Stanford into a ferroptosis-resistant U2OS cell line. And here we're simply looking for dropout. If we've knocked out a factor that's protecting these cells, they'd be depleted from the final population. We can deconvolve this by deep sequencing. Now, I remember when we did this screen, we had all kinds of ideas. We were going to get lots of hits. How are we going to prioritize these, putting them into different pathways? And then we did the screen, and it was pretty clear what we should prioritize. This gene up here had a really high confidence score and a strong sensitizing effect, as previously known as AFM2, and we renamed this as ferroptosis suppressor protein 1, or FSP1, and this was co-discovered in parallel by Marcus Conrad and Pedro Angeli at the Helmholtz Institute in Munich. For the sake of time, I'm just going to summarize a lot of the mechanistic understanding of FSP1 here so I can tell you about some of our more recent studies. What we found is that FSP1 is an oxidoreductase. It localizes to the plasma membrane, and what it does is that it uses NADPH or NADH to reduce coenzyme Q10 to its antioxidant form. So this can function as an antioxidant similar to things like vitamin E. And what was perhaps surprising here was that this was using non-mitochondrial pools of CoQ. So this solved a long-standing mystery in the field of what CoQ does outside of the mitochondria. Our data suggests that it's there to enable and prevent membrane lipid damage. And now this raised several questions that we've been interested in addressing over the last few years. One is this concept of lipid quality control. I think that this was an initial example of how we can protect the plasma membrane from damage, but you can imagine your favorite organelle, lysosomes, peroxisomes, mitochondria, ER, each of these have distinct lipid compositions, protein compositions, and are susceptible to different redox environments, and likely need different mechanisms to protect their lipids. So I think we're kind of scratching the top of the surface here. I think we've been very interested in regulation of FSP1 expression and stability and how this relates to cancer therapy resistance. And then we've been interested in developing and targeting FSP1 for therapeutic benefit. And I'll try to touch on each of these over the next 10 or 15 minutes. So in terms of thinking about subcellular lipid quality control, we immediately got a clue from the localization of FSP1. In addition to being present at the plasma membrane, FSP1 very strongly localizes to lipid droplets. And so this led to a very clear hypothesis that a current postdoc in the lab, Mike Lange, has been studying, which is that FSP1 can protect stored neutral lipids, like triacylglycerol and cholesterol esters, from oxidative damage. So lipid droplets, you may be familiar with these organelles. They're ER-derived neutral lipid storage organelles that emerge from the outer leaflet of the ER into the cytoplasm. And they really function as dynamic hubs of lipid metabolism. So they can be access to mobilized fatty acids that can be used for energy, for making new membranes, lipid signaling molecules. And they can also play roles in terms of lipotoxicity, where the idea is they can sequester potentially toxic lipids. And this is the idea and concept in the ferroptosis field, that these lipid droplets can sequester PUFAs, thereby preventing their flux into membranes and protecting those membranes from oxidative damage. But I think this idea raises a really important question as to what is the fate of those PUFAs upon incorporation into the droplet. Are they somehow protected from damage in this organelle? And so what Mike did is he generated FSP1 knockout cells, and then together with Maria Fedorova in Dresden, performed a series of lipidomic experiments. And I just want to show you one here that was really striking. So what you see here in this volcano plot are oxidized lipids that accumulate in the FSP1 knockout. And this was really exciting for us because this is in the absence of any other kind of ferroptosis inducer, for example, a GPX4 inhibitor. And what we see is these yellow or orange circles. These are all oxidized triacylglycerol molecules that are accumulating in the lipid droplet in response to loss of FSP1. We could also target FSP1 directly to the droplet, and this is sufficient to suppress this damage, suggesting it's playing a direct role in this process. Similarly, we can reconstitute this, so we can make artificial lipid droplets in vitro. This would be similar to a liposome, but in this case, this is a droplet with neutral lipids in the core. We can initiate lipid peroxidation, and we have a fluorescent readout, this BODIPI-C11, of lipid peroxidation. What you can see is when we add FSP1 together with its substrates, we get this lag phase or suppression. And what happens at this inflection point is FSP1 has consumed all the NADH, so it can no longer recycle CoQ. If we add more NADH, we just extend that lag phase longer and longer. So in vitro, in this biochemically reconstituted system, FSP1 can directly suppress neutral lipid damage. So we wondered about the physiological consequence of this, and we can take advantage of the fact that lipid droplets can be de novo synthesized, so we can manipulate the composition just by adding different types of fatty acids. For example, we can add oxidation-prone PUFAs and initiate the formation of a PUFA-rich lipid droplet, we can then inhibit FSP1 and accumulate oxidized neutral lipids and look at the downstream consequences. So when we add PUFAs and initiate this highly prone, oxidation-prone lipid droplet, up until about 200 micromolar, our wild-type cells are completely fine. But we see something quite different in our FSP1 knockout. Even at these earlier concentrations, we see that these cells start to die. And by about 200 micromolar, these cells are all dead in the FSP1 knockout. And this requires the formation of these PUFA-rich lipid droplets. So we see this death that's happening in the FSP1 knockout. It's actually worsened if we inhibit HEGL, the rate-limiting enzyme in lipolysis, and it's completely blocked if we inhibit the DGATs, which are required for the synthesis of triacylglycerol, indicating that these droplets are required for this effect. So what this data tells us is that in addition to playing a role at the plasma membrane, FSP1 is also acting on lipid droplets to protect stored neutral lipids. And I think that this is very important because the field now probably has thousands of papers on phospholipid oxidation, but now this also tells us that these stored neutral lipids can be oxidized. And I think this is going to be important in many diseases like fatty liver or during aging in glial lipid droplets. These are some areas we're interested in studying. So this establishes a really good exemplar for a compartment-specific lipid quality control mechanism where FSP1 is recycling stored neutral lipids, stored antioxidants to protect neutral lipids. And it also indicates this additional emerging idea in the ferroptosis field that you can have ferroptosis being initiated at different intracellular membranes. And so I think ferroptosis isn't one pathway. We can have initiation from multiple organelles, and this propagates to the plasma membrane. And I think this is also a major question of how this actually propagates. So we're very interested in thinking about FSP1 and how it's regulated. And Karen Deep-Diol, now a senior scientist at Genentech, wanted to understand the regulation of FSP1 abundance and how this determines cellular antioxidant capacity. And we again turned to genetics, and what Karen Deep generated here is she knocked into the endogenous locus a GFP P2A BFP tag. And this is useful because it provides a reporter of FSP1 levels, a reporter of FSP1 transcription or expression, and also by looking at the ratio, this provides a reporter of FSP1 post-translational stability. So we can uncouple these effects on its abundance. So Karen Deep then did several screens under different conditions, both for the transcriptional regulators, sorting with BFP, or for post-translational regulators, sorting for this ratio. And we saw many things we'd expect, for example, NRF2, which is involved in the transcription of FSP1. But we also identified this pathway. These two genes came out very strongly. When knocked out, we destabilized FSP1. So RFK and FLAD1, these mediate the conversion of riboflavin or vitamin B2 into FAD. And FAD is incorporated into FSP1, so it's an FAD-dependent oxidoreductase. But this suggested that not only is it important for its activity, it's also important for regulating its stability. And similar findings I just want to mention were found by Pedro Angeli's lab. So indeed, if we knock out RFK, we see this destabilization in FSP1 levels. So a pretty profound shift here in the fluorescence histogram. And if we look at the half-life, we have a strong shift from the wild type, which is quite stable, around 20 hours, to about a 6-hour half-life. So it's destabilized. Now, when we knock out RFK, it's broadly affecting FAD levels in cells, and there are many different FAD coordinating enzymes. So we really wanted to understand if this is direct. And so we're able to take advantage of recent structures that have been solved for FSP1 and make direct mutations to the amino acids that coordinate FAD within FSP1. So in this case, we would only be affecting FAD binding by FSP1, not more broadly. And indeed, similar to when we knock out RFK, we see this dramatic effect on FSP1 stability that's recapitulated here. When we just make these mutations, and we see similar with findings with other mutations to FSP1. So what this data suggests is that when FSP1 is unable to bind to FAD, that it's rapidly recognized and degraded. And in data I didn't show, we've defined the E3 ligase pathway. It's mediated by RNF8. I think conceptually what is important about this work is it suggests that vitamin involvement in ferroptosis goes beyond simply radical trapping antioxidants like vitamin E and vitamin K to also being important for regulating FSP1 activity and stability. And this also suggests something very interesting to us, that dietary modulation of FSP1 might be possible. In line with this, there's a really interesting paper from Misha Jain's lab at the Gladstone at the ARC just out last month, in which she showed that in this GPX4 knockout model, they're sensitized by a vitamin B2 deficient diet. So I think our findings are in very good agreement with this, that perhaps in these vitamin B2 deficient diets, you have a defect in FSP1 stability and activity. So we might be able to target FSP1 with diet, but we might also be able to directly target FSP1 with small molecules or other approaches. And this is work from a previous graduate student, Joe Hendricks, and a current postdoc, Amalia Magarodi. Can we just directly target FSP1? And we are motivated by findings from other groups in the field. For example, this paper out in the fall from Thales Papagiannakopoulos' lab, in which he shows that if you knock out FSP1, you have a strong reduction in tumor burden. And this was really exciting for us to see, because if you knock out FSP1 in vitro, these cells will continue to grow, but they're highly sensitized to ferroptosis. But the situation in vivo is very different. And here we see that knockout of FSP1 alone is sufficient to reduce tumor burden. We've also seen, this is a collaboration together with Michael Dupage's group, that if we knock out FSP1 in T cells or just in Tregs, we also get a strong restriction in tumor growth. And this suggests that we might get synergy between targeting FSP1 in the immune system and targeting FSP1 in the tumor. So with this in mind, we went forward and developed small molecule inhibitors. This was a collaboration with Julia Schletsky and Eddie Weary at the UC Berkeley Drug Discovery. We screened 115,000 small molecules and identified several chemical scaffolds that we think will be useful moving forward. We've primarily followed up mechanistically on this first molecule, FSN1, but we're continuing to do medicinal chemistry on some of the other chemical scaffolds as well. This molecule has a respectable IC50, around 300 nanomolar straight out of the deck, and we're working to improve potency here. And we also see very nice induction of ferroptosis in this very aggressive lung cancer line when we inhibit FSP1 together with the GPX4 and RSL3. So we see near complete induction of ferroptosis in this experiment. Now this experiment also led us to several mysteries. We noted that FSN1, our FSP1 inhibitor, was selective to human FSP1, so it didn't inhibit mouse FSP1, and we didn't understand the mechanism at this point either. So we were fortunate to collaborate with Da Jia and Sichuan University in China to solve the co-crystal structure of FSN1 together with FSP1. And what we saw was quite informative. So FSN1 here, this molecule buries in this pocket and makes key pi-pi stacking interactions with this phenylalanine 360. And actually this phenylalanine in mouse is a leucine, which solved the question of why this is human-specific. So we can mutate the phenylalanine to a leucine, we lose the ability to inhibit, and we can also humanize our mouse. We can mutate the leucine to a phenylalanine, and then we can inhibit the mouse version with our molecule. So I think this will be useful moving forward as we begin to move into preclinical models. So I want to end here just by summarizing. What I told you today is how we are about our discovery of FSP1 and its mechanism of action. It acts by recycling a lipid antioxidant, CoQ, and suppressing the propagation of these lipid peroxides in the membrane. In addition to functioning at the plasma membrane to suppress ferroptosis, it also functions at intracellular organelles like the lipid droplet, where it protects stored neutrolipids from damage. And I think that conceptually, this is a really good example of subcellular organelle-specific lipid quality control mechanisms. And moving forward, this is an area we'd really like to explore. How does the ER protect its lipids? How do mitochondria protect their lipids? And how do other organelles protect their lipids? I told you about a news story in terms of regulation of FSP1 levels and about some of our efforts to develop small molecules to therapeutically target FSP1. And I think FSP1 might be a very good therapeutic target with a very strong therapeutic window. For example, GPX4, which a lot of companies have gone off after initially, is actually really challenging to target because it's so essential in other tissues. It's embryonic lethal if you delete it. Postnatally, you get kidney degeneration, but knockout of FSP1 is really well tolerated by mice, which I think is indicative of a strong therapeutic window for FSP1 targeting therapeutics. I'd like to end by thanking the members of my group who I tried to point out along the way. Again, the Lipid Research Division here at ASBMB, and also our funding sources. And I just want to also thank more broadly the lipid cell biology community, my colleagues at UC Berkeley, and also my family here. Thank you very much. I'm happy to take any questions.